name is Barrett Carson, and I'm in my 25th year as vice president at Georgia Tech. I bring you the remnants of my morning coffee. Let's move it a little bit closer for a little product placement. Waffle House coffee, Arabica beans selected from the hilltops of South and Central America, brought to the United States and gently roasted and blended over in Birmingham, Alabama. So I want to thank the folks from Waffle House, one or two of which may be on the call today, and also especially Joe Rogers, Bert Thornton, and Walt Amer. And I learned in the process that they actually have those Keurig cups of their coffee in there, and so I'm going online now that I finished my first batch and ordering some more. So I urge you to join me in there, wafflehouse.com. I've also been accused of having a fake library behind me, and as probably the only history major here on the call, you can't do fake in history, and so the only way I can think to prove it is to reach back live. Oh, look what's there! A little Coca-Cola, and let me get the meeting started with that. Whoa. Well, okay, it will come out. Well, anyway, and we drink this stuff by the case when everybody else was out getting down the paper aisle in the local Publix in there. I hit the Coca-Cola aisle. Can't live without it. So again, we have 500 million reasons to say thank you to Coca-Cola and the generosity of Letty Pate Whitehead Evans. We now benefit each year from about $14 million, all generated from the initial generosity of Coca-Cola stock. I want to welcome all the members of the Hill Society who are here with us today, as well as members of the Georgia Tech Foundation Board of Trustees and the Georgia Tech Advisory Board. Welcome each and all. This is the next iteration of Conversations on the Hill, directed to all of our guests in there, and we have an extraordinary program here today. Let me introduce now the 12th president of the Georgia Institute of Technology, Angel Cabrera. Angel is a native of Madrid, Spain, and a graduate of the Polytechnic University of Madrid before coming to North Avenue to pursue his master's and PhD degrees, where he met his wife, Beth, and from Florence, Alabama, and they actually sent on their son, Alex, to Georgia Tech as well. So they're Georgia Tech dual alumni as well as parents. Angel is, I first met Angel when he was president of the Thunderbird School out in Phoenix, and recruited him on the spot to join the Georgia Tech Advisory Board, where he served 2 terms and became chair before he moved on to the George Mason University up in Fairfax, Virginia, where I had the honor of representing Georgia Tech at his inauguration. Angel joined Georgia Tech in September of 2019. So he had about 7 months on the ground before the entire world closed down around him. So right now we're trying to get him out, beaming him out, since we can't get him out physically for another couple of months in there. With that, let me turn it over to Angel Cabrera, Mr. President.
Thank you. Thank you so much, Barrett. And what a pleasure to share this morning with you. And as you will see, we have this is an amazing duo that we're going to be hearing from today. It is, I think this year has made it very, very clear how essential it is that we continue to invest in science, in research. We've seen it, of course, in the context of COVID, and how the research that is conducted in places like Georgia Tech has really helped us deal with this unprecedented pandemic. But there is a lot more to be solved beyond COVID. And of course, the disease that occupies this conversation today is one of those, is Alzheimer's. It is one of the leading causes of death in the U.S. There are an estimated 5 million cases in the U.S. and about 50 million cases worldwide. It just causes just incalculable personal and family pain, and also, of course, incalculable economic damage. It's a very complex disease, and science has yet to determine how to best treat it, and while there are families that are affected by this that are waiting progress, and you should be very, very proud that the research that is conducted right here at Georgia Tech through our bioengineering program, and very particularly through our culture department of biomedical engineering in partnership with Emory University, also through our neural engineering center. I mean, there are different units through our campus that are doing cutting edge research and very exciting research that to help us understand and treat this disease. And I couldn't think of two better people to share with us what that research is showing us, and to help us understand a little bit more the complexity of this disease. And the first person I'm pleased and delighted to introduce is Susan Margulis, who is the chair of the Department of Biomedical Engineering at Tech and Emory. As you know, this is a unique partnership between two universities. The only case I know where a department really is owned and by two different universities. It involves nearly 70 faculty, over 1,000 undergraduates, and hundreds of graduate students. Is ranked right now #2 in the nation by U.S. News and World Report. It's absolutely an incredible program. Now, Susan herself, she has had an incredible career in biomedical engineering. She's an expert in, among other things, in traumatic brain injuries. And in 2020, even though it was a tough year for all of us, at least it had a little bit of a silver lining for her, because she was named in the same year. She was elected to both the National Academy of Engineering and the National Academy of Medicine. And I don't know how many times that may have happened, if at all, for one person to be elected in the same year to both, which is a huge career milestone, and we're incredibly proud of her. And she's going to be today's moderator. And then, of course, our featured speaker is Dr. Annabelle Singer, who is a professor in the Department of Biomedical Engineering and joined us in 2016. The goal of her research is to understand how neural activity produces memories and spurs the brain's immune system. She integrates behavioral, electrophysiological, and computational methods to identify and restore failures in neural activity that lead to memory impairment. She's just using incredibly innovative approaches, and I will shut up now and let you hear from them. This is incredibly proud to leave you with Susan and Annabelle. You have the floor.
Thank you, President Cabrera. It's my pleasure to moderate today's session with a very, very special member of our faculty. I want to begin with just a few event details. Realize that the event will be recorded and that link will be sent to you. So if there are people that you want to share this with, please do. And to remember to mute your microphone during the event. I actually had to stop a neighbor from mowing her lawn next door and asking her to refrain just a little bit. Questions, and I'm sure there will be some questions, should be submitted in the chat. And the chat looks like one of those talking bubbles from a cartoon up at the top of your screen. And if you click on it at the bottom there, you can just type in a message. You don't need to wait till the end, and I'll be looking for those messages. And at the end, we'll be sure to leave time for your questions. And I look forward to your questions. So I'm going to take us today on a journey inside the research and the career path history of Dr. Annabelle Singer. So I'm going to begin with a series of questions that will really help us understand, what is she studying? What is she finding in those studies? and why is she studying this, and the directions that her research will take in the future. Annabelle, thank you very much for being here.
Thank you for having me.
I wanted to begin with, how did you become interested in studying Alzheimer's disease?
Yeah, I mean, there were many steps along the way, but probably one of the early profound ones was when I was in grad school. I was doing really basic science research on memory, but I was very interested in disease. And I asked the Memory and Aging Center there, so the premier center that evaluates patients, if I could shadow their neurologists, simply for my own curiosity. And they were really kind in that they let me do that. And it was an incredibly powerful experience. I can still remember vividly the patients that they saw, their families, how the families came together to support somebody going through these devastating diseases. And it really struck me how little the neurologists had to offer. The things they had to offer those patients was a clear diagnosis, which was a relief for the patients and the families, but that was almost it. They had very little to help those patients. So that really stuck with me, and still to this day sticks with me, and that there's a huge need in this community.
So what brought you to our department at Georgia Tech and Emory?
Yeah, so really a conversation with my friend Sam Sober, who's faculty at Emory. I talked to him, I told him I was, looking to apply for faculty jobs, and he really encouraged me to apply to the BME department at Georgia Tech and Emory. And he told me it was a place that was doing really incredible research, but also was a wonderful place to be. I would have wonderful colleagues, and it would be a great place to develop as a faculty member. And then I applied. When I interviewed, before I had my on-site interview, I had an hour-long phone call with Lena Ting, who's one of the members of the search committee and one of our faculty members. And she gave me advice about how to do a good job, which I was supposed to be impressing Lena, right? I was supposed to be impressing the faculty, and instead I was getting support and mentorship about how to be a star. And that really stuck with me as well. I thought, wow, this is a place where I could grow, where people are going to invest in me and how I can be the best at what I do. So those two really made it key for me to come.
That's great. So now let's jump right into your research. So your research has traditionally been in mice, and then more recently now in Alzheimer's disease patients. Tell us a little bit about your exciting research.
Yes. So really, actually, I started in rats, and I studied neural activity and how neural circuits that we know are important for memory produce these memories. And I knew I wanted to apply this to Alzheimer's. At the time, that was a really unpopular thing to do. Most Alzheimer's research focused at that time, and still does, focus on what we call the molecular pathology of the disease. So things like amyloid beta that forms plaques, or phosphorylated tau that forms tangles. But I wanted to study how neurons behave in the disease, how they fail so that you get this kind of memory impairment that is a hallmark of the disease. And I brought this to my advisor at the time at Boyden, and he said, well, so what, who cares? How's that going to lead to a therapy? So, I racked my brain on that. So we started by looking at these kind of neural activity and how it fails. And we found a deficit in a pattern of activity we call replay. You can kind of think of it like a sports replay, where in a sports replay, there's the actual play, and then you get a snippet of the key parts that get replayed. We see the same thing with neural activity. When an animal gets a little reward, you see replay of the path that they took to get that reward. And that was a major part of my PhD. So I hypothesized something was going wrong with this replay activity in the Alzheimer's mouse model. So these are mice that are programmed to develop Alzheimer's. And in fact, that's what we found. And that was really, to me anyway, potentially linking how we get from the molecular pathology to the memory impairment. But I still have this question in my mind, how do we turn this into a therapy? So what? Where's the end goal here from my advisor? And so we decided to try to re-inject or engineer that activity to bring it back as a kind of rescue. And that's what we did. So we had to look at that activity, figure out what we could bring back, and that was a pattern we call gamma oscillation. So gamma is a pattern of electrical activity in the brain that's been studied for decades. And we think it's involved in a variety of key functions, including memory. And we knew we could stimulate at gamma. So gamma is around 30 to 50 hertz. We knew we could stimulate right in the middle there at 40 hertz. And so we did that in our mouse model. And we found, lo and behold, by driving this activity that was missing, we could reduce the molecular pathology of the disease. So we saw a reduction in amyloid beta. Later, we saw a reduction in phosphorylated tau. And we really surprisingly were changing the brain's immune cells. So there are these cells called microglia. They're kind of like the trash collectors of the brain. And normally they just sit there at rest. But when we stimulated, we changed them to do their job. So they would then move around, they changed their morphology, and they would go around and pick up the amyloid beta and clear it out. So we were able to go all the way from this deficit to a new kind of stimulation that's potentially therapeutic. And then we wanted to know, well, how can we do this in a way that we could translate to humans? Because that initial stimulation was very invasive and not something that currently works in humans. So we actually tried a number of different things. And interestingly, the thing that worked was what we call sensory flicker. So if we simply turn on and off lights and sounds at a particular frequency, in our case, 40 hertz, you can entrain that electrical neural activity at the same frequency in the brain. We knew we could do this with what we call sensory brain areas, so brain areas that are important for processing vision or hearing. But what we found is that flickering lights and sound, we could entrain the memory circuits that are important for Alzheimer's disease. And if we do it for long enough, in mice, that was an hour a day for 7 days, we could also reduce this amyloid beta and phosphorylated tau and transform these immune cells in the brain.
So let me just make sure that I'm understanding it, and for others as well. So you found that this gamma frequency was at a lower level, or maybe even missing, in animals that had, essentially, they were models of Alzheimer's disease, and that by putting it back in, you basically didn't just get back to normal, but even ramped up or amped up the garbage collector cells. That, and then also, did you find improvement in memory at the same time?
Yes, absolutely. So once we knew how to do this, where we could target memory circuits with this non-invasive sensory stimulation, we looked at behavior. And in fact, we were rescuing what we call spatial memory behavior in the mouse models.
Remembering where you were and how you got there.
Exactly.
So, and the second piece I want to make sure that I understand is that those early studies were putting electrodes into brains, and you can't really do that with patients. So next you figured out, much in the same way when you are on a Stairmaster or a bicycle or walking, your breathing actually gets entrained with the pace of your legs, and that you basically found that by using light and sound, you were creating those wavelengths that you needed, that you had been using needles directly into the brain to create. And the light and the sound alone, without any needles at all, was making that frequency happen.
Exactly. I love that analogy of your breathing sinks into what you're doing. Yes, we think the neurons are sinking into this sensory input.
And so did you try using this in animals before they had those plaques? Because it gives kind of the opportunity you talked about as a treatment. And Alzheimer's disease is not a moment in time. As we know, and as many of the people on the call know, they're early stages of the disease. When it's suspected, there are none of these tangles in the brain yet. They're just early signs. So in your animals, did you study different stages of the disease?
So in those initial studies, it was really what you might call mid stage of the disease, or somewhat early, but so we studied in animals that had memory deficits. We also studied a little bit earlier than that when you already see plaques and amyloid building up. We're now looking at ways that we can use this stimulation preventatively. And that's really a whole undertaking in and of itself. I get asked all the time, can we all just use this? Can we just start stimulating now? You know, pre-symptoms, no idea if we're even at risk, but just do it. But there's potentially downsides, and so we have to study that explicitly. And we're studying that now in animal models, and then we'll translate that to humans as well.
In some of your research, you found even in the animals that if you change the frequency and do another frequency, it doesn't have a positive effect. It may even have. I'm not sure what that is.
Yeah, we're gonna. If anybody isn't muted, go ahead and mute yourself. That should help with the feedback. Yes, so so in some of these studies where we're looking at using the stimulation preventatively, we are trying different frequencies of stimulation, and that is informed by studies we've done where we find different frequencies of stimulation have different effects. That's really, if somebody's not muted, go ahead and mute yourself. So the different frequencies of stimulation have different effects, and that's potentially really useful in a variety of different reasons. And one of them is to tune the stimulation to the individual. And we're looking at that now in terms of trying to prevent developing risk for Alzheimer's. One of the key risk factors of developing neurodegenerative diseases, and Alzheimer's in particular, is chronic stress. So we're trying to prevent the pathological effects of stress. And we're finding some really interesting initial effects there that we think are, we're seeing the behavioral effects, and we're picking apart the mechanism right now. But it matters how you do the stimulation, and it differs depending on the sex of the animal. And we think ultimately in humans, it may differ depending on the particular individual. So ultimately, the way we use the stimulation may be that we take an initial immune profile of the individual and then tune the stimulation to that particular individual.
In a way, we do that now with hearing aids. It's not one size fits all. You don't just amplify the sound across the entire spectrum. Certain frequencies are up higher than others depending on your particular ears. So, exciting research in animals, but now share with us about the transition that you've made to study humans.
Yes, so after we had this non-invasive form of stimulation, I knew that opened us up to try this in humans. It was non-invasive, it's something that we could reach memory circuits, and we could have all these beneficial effects. So I went to Alan Levy, who's the chair of neurology at Emory. I gave him an update about where we were with our research, and I asked if we could test it in humans. And in fact, I expected him to tell us 10 years of additional preclinical studies that we would have to do. I was, I thought it would be 5 years at best of more studies we had to do. But instead, what he said was, yes, let's do it. Let's try it. Let me introduce you to our study staff who make these studies happen. And so we did. It was really a huge opportunity for us to quickly take this from what we call bench to bedside. And we just completed that first study a few months ago.
And what were the challenges that you found, the two biggest challenges? And if I could say, what surprised you most?
Yeah, so it's a huge challenge, taking things from a mouse model to humans. It's hard to describe how different they are and how many decisions you have to make along the way. So the study team and I met every week for weeks on end, discussing every part of what we had done in mice and how that would be applied to humans. So for example, in our mouse studies, we did an hour a day of stimulation for 7 days and saw beneficial effects. Would that be enough in a human? Well, our first guess was that would not be enough, so we decided to do it longer. but we didn't want to go so long that we would have to wait years to get the results. So we settled on an initial study that would be 4 to 8 weeks long with a extension that goes out to a year. And should it be an hour a day? Should it be a half hour a day? Should it be two half-hour sessions a day? All these things were open questions. And we had to make some decisions in terms of what we knew based on mice and how they relate to humans. Even every assay that we do in mice, you can't do it in humans. The most of them are too invasive. So every assay, we had to figure out what is an equivalent that measures the same underlying biological process, but can be done in humans and would hopefully be sensitive enough in our human patients. Another key challenge was clinicians were really doubtful that patients would do it at all. When we described the stimulation and tried to show it to them, they thought nobody would want to do this stimulation, that we would never enroll a single participant. And if we did manage to enroll somebody and we then sent them home with a device, there was no way they were going to keep doing it day in and day out. We have trouble getting patients to take a drug every day reliably. So how could we ask them to do an hour a day of this stimulation?
And you were asking people to wear goggles and put headphones on or something? Okay.
Yeah. So there's goggles that have flashing lights and then headphones that have a kind of fast beeping sound. It's like the lights are like a fast strobe light and the sound is like, it's faster than most beeping.
And you were asking them to do nothing else while they did that?
Yes, exactly. So they have to just sit there and do it for an hour. And so you asked about the surprises. Well, one of the big surprises was that the adherence and the tolerance, the technical terms we use, were really excellent. So people were fine with the stimulation. I think of all the people we screened, one person didn't like the stimulation, but they were excluded from the study for other reasons. And then as far as adherence, participants did it and they did it reliably. Most participants did more than 90% of their prescribed treatment time. And I think the lowest was 89%. On average, it was something like 95%. So those are really high adherence rates. That showed us that we had figured out how to get participants to do it reliably. It's not just that we handed them a device and sent them home. We actually did a lot in the study to make it so that they would keep going and be motivated to keep going.
So what did people say about the experience? What was some of the feedback you got from these patients?
Yeah, that was really interesting. So participants did say at first it was really hard to find time to do it. So the participants that we were working with are what we call prodromal Alzheimer's. They have prodromal Alzheimer's disease. So they're early in the disease. A lot of them still have active lives. They're not sitting around all day. So a lot of them, or some of them anyway, said it was hard to fit in an hour of doing this every day. But they figured out how to do it, and it became a kind of special time for them. Maybe even meditative, or a me time was what one participant said. So that was really enlightening, and actually maybe some of the beneficial effects will be getting people to spend a little bit more me time in their busy lives. That was one thing that they said. We got a lot of feedback about the details of how the device worked and things like that. One of the other things that happened is at the end of the study, we had a meeting with all the participants, and I was struck by how grateful they were. Even though they knew it was an early stage device, we were not pitching it to them as something that would benefit them. They were doing something to benefit society, really. But they were really grateful. They, I think, were grateful about the research we were doing, but also they were very hopeful that it was beneficial.
So for this group, how big was the group? What age range was the group? Men, women?
Yeah, so we had 10 participants in this first study, men and women. I think they are all over the age of 65, but I'd have to go back and check.
Good. And so men and women?
Yes.
Okay, good. So did you end up finding that the human brains responded to the gamma exposure like the mice?
Yeah, so this first study, the main outcomes were what we call safety, tolerance, and adherence, which were all excellent. But we also looked at a variety of what we call biological outcomes that would tell us not conclusively that it's efficacious, but what would be preliminary evidence, because it's a small study and it's short. So it's preliminary. But we did find some really intriguing preliminary results. One of them is that we see changes in brain network connectivity. So this is how brain regions talk to each other. And there are particular brain networks that are involved in memory that are weakened. The way they talk to each other is weakened in Alzheimer's disease. And we saw some strengthening after 8 weeks of flicker in those networks. And so that was really intriguing and something we'll be following up on in a bigger study. We also looked for signs of immune effects. So we can't measure microglia the same way in humans as we did in mice, but we can measure immune signals called cytokines that are present in the brain, but also in the cerebrospinal fluid. And we looked at those cytokines in mice, and we then looked at them as well in our human participants. And we did find significant changes in what we call a cytokine profile. So we don't just look at one cytokine, we look at 40 at a time. And we saw a very different profile in participants after 8 weeks of flicker. And in particular, we saw some cytokines went down that are regulators of the immune response. So that to us indicates we might be getting a kind of regulation of the negative immune signals or inflammatory signals with flicker. Both those things we're going to be following up on in larger and longer trials coming up.
That sounds great. So what are the steps from this type of a study to really reaching patients all over the nation?
Yeah, so first it'll be via clinical trials, and subsequent trials as they get bigger, they become multi-center trials. So there will be trials beyond Emory in the future. And beyond Georgia Tech. And then after that, hopefully it is a FDA approved medical device that you could get prescribed by your doctor. Realistically, I know there are already people who are using, they home built their own device and they're using it. I can't recommend that outside of the supervision of a physician, but it is happening.
That's interesting. So it's simple enough that somebody could, build this in my garage kind of thing. Interesting.
It's very simple, especially if you have an engineering background.
So what are some of the directions that you plan on pursuing in the next few years?
Yeah. So one of the big ones is bigger, longer trials, as I just mentioned. But beyond that, we are aiming to apply the stimulation to other neurological and psychiatric conditions. So as I mentioned, with different forms of stimulation, we get different effects. We can increase cytokines, we can decrease cytokines with different kinds of stimulation. And we're now looking at some more subtle changes. Can we optimize a particular signaling pathway that's for a particular disease, for example? What that means is that we now have a really flexible, non-invasive tool to control brain immune function. That's useful in a lot of diseases. It's useful for Parkinson's potentially, where we think inflammation is playing a role in why neurons die in the disease. It's potentially useful in ALS. Again, we think inflammation is playing a key role. It's also, those are both neurodegenerative diseases. It's also potentially useful in psychiatric diseases like depression and anxiety disorders. There's a big immune component to those. And there's evidence that there's an immune component to diseases like schizophrenia and autism. So all of these different diseases, being able to manipulate the immune system could lead to a new therapy. With each disease, we have to figure out what's the kind of manipulation we need. So we can't just assume it's going to be 40 hertz in all cases. And then one of the other key things that we talked about already a little bit is trying to use the stimulation to prevent disease onset. So things like if we can prevent the pathological effects of stress, that would lead to a drastic reduction not only in Alzheimer's disease, but also in major depressive disorder and anxiety disorders. So it's a huge risk factor for a variety of different diseases.
So I want to share a little bit about sort of how research happens, and then ask you to explain where your ideas come from. So research is sometimes funded research, where you write a grant, you have some preliminary evidence, and then you submit your ideas for review of other scientists, by other scientists, and with funding possibly coming from the National Institutes of Health, or perhaps the Department of Defense, or the National Science Foundation. But you've just named a whole bunch of diseases that you have not even studied. So the other place that scientists go is serendipitously, or sometimes in a more planful way, funding comes that is wide open blue sky money. And you received, in my very first year, one of my very first kind of official things was was literally to surprise you. The Packard Foundation called me and said that you were about to receive a Packard Fellowship, which is a very distinguished Genius Award. And they basically wanted to call you and surprise you. And my task was to have you be with me and be surprised. And it was a very interesting request, very unusual. So we had lunch together on Fine China in my office. And during that lunch and before your phone call, I asked you, what would you do if you suddenly had $1 million, and what you would do with that? And you started talking about ideas that were these critical blue sky kinds of ideas. I wanted you to share with us a little bit about how you pick the questions, how you decide what you're going to study. At the very beginning of today, you said you watched patients, but tell us a little bit about where those ideas come from of what you're going to study next.
Yeah, that's a great question. I have a ton of ideas, and I would say probably most of them come from when I'm talking to people. Working with people, it's one of the wonderful things about research is it's really a social endeavor. And when we're talking and sort of having fun with research, we're thinking big, right? And that's something actually we do in my lab. Every few weeks we have a lab meeting that's about our dream big ideas. So I try to also instill that in my trainees. I also try to set aside time for brainstorming. That's really important, and maybe I need to do more of it. As things go along, I get busier and busier. But it's really important to see the long-term goals, not just the small short-term goals. But then, okay, so I have a ton of ideas. I have never really had a lack of ideas. Then the issue comes, how do you pick among them, right? Because you can only pursue so many. And one of the keys there is figuring out among the most potentially impactful ideas, which ones are feasible. And one of the ways that we try to do that, so in my lab, I like to give people the opportunity to take a big risk, right? Dream big, take the risk. But how do we do that in a way that won't, sink all of our resources or their careers and their mental health? Well, we do that by what I call de-risking fast, and then we pivot if it's not working. So de-risking fast is, I literally draw a chart for my lab where it's like on the x-axis is time and on the y-axis is risk, and you start way up here, right? High risk, you haven't spent any time. Feels good. You have no idea what you're in for. What you really want to do is get way down on the risk axis as quickly as possible. And so we think about what's the most likely reason that this project is going to fail, and how can we test that as quickly as possible. If it fails, no problem. You didn't spend much time on it. You pivot and you try something else.
Yeah, these are the killer experiments. You just want to know whether to kill the study and the rest of the direction. So this is a long haul. At Thanksgiving, people say, you're still studying brain injury. I can't believe you haven't, solved that one yet. So I'm curious, what energizes you in this long journey?
Yes, it's definitely a marathon. What energizes me, I would say, is working with trainees and students, they're really wonderful. They're full of energy. They are full of hope. They have none of the cynicism that we develop after years of, rejections and failed experiments. And that is huge. I love working with students. Another big part for me has been working with patients, seeing the end game, right? Our end game is really to make a difference in patients' lives. And when we're working in the lab, that feels really far away. So getting to interact with patients or families of patients has been really helpful and re-energizing for me, especially in this pandemic. Hearing from patients and hearing their hope has been really helpful.
Thank you. So there have been a couple of questions in the chat that I'd like to weave in now. One of the questions was, when you start studying other diseases, do you think you always have to start with the animals, or would you ever start with people?
Yeah, that's a great question. Well, always is a very strong term. So right now we're starting with the animals, and there's a good reason to do that, because we can do a lot more with the animals more quickly than we can do a human trial. Human trials are a huge undertaking in terms of time and money and the setup, the regulatory setup. So doing an animal study is faster, almost always across the board. But there may be times when there isn't a good animal model, in which case we might not be able to do an animal study first. And overall, we're finding the safety of the stimulation, at least in the human studies we've done so far, the safety has been really good. So that would be a good rationale for just trying it in another patient population without doing more preclinical studies.
And I would add that there are things that you can do at the end of an animal's life that you're never going to do to get that information in a patient that is invasive, that you may have to sacrifice the animal and look at tissues that you couldn't get otherwise. So it's important to do those animal studies to understand not just whether it's working, but how it's working too.
Exactly. We can only really get at mechanism through animal models. And in addition, the animal models are faster, not just because of the regulatory component, but they're often, they age faster. So diseases typically progress faster in the mice. One of the big challenges in Alzheimer's disease research is it takes a long time for the disease to progress. So it takes a long time to see effects, versus in the animal, we can see that progression is over a couple months.
So Roxanne Westerhoff has an interesting question. So you mentioned that stress can be something that is present, especially prolonged stress, in Alzheimer's disease. And she asks, what about other diseases that are not brain diseases, that are known to connect with stress, like cardiovascular disease? Do you think there may be a role for this type of therapy, maybe gamma, but at least the flicker therapy for cardiovascular disease?
Yeah, that's a really interesting question. So I haven't thought about that from the stress angle, but I've thought about it from a different angle, which is our nervous system innervates every part of our body. And there are people right now working on what we call electroceuticals, which is using stimulation of the nerves to control organs that are not the brain. For instance, for treating diabetes by controlling the pancreas. Pacemaker is a kind of electrical stimulation, right? So it's possible that there would be some beneficial effects there. I haven't thought about it through the lens of stress, though, before, and I'd have to actually look at what the key components are of that effect.
Okay, good.
There's a potential there. The stress effect does come to our brain, right? The release of stress hormones is regulated by the brain.
Mm-hmm. So your research has certainly benefited also from generosity from Georgia Tech alums. And I was wondering how that connection came about and what it means to you to actually to interact with the alums.
Yeah, great question. So yes, my research has benefited tremendously from a group of alumni. And that started with a meeting. So Steve Cross introduced me to Alan Ecker, who's a Georgia Tech alum. And we had a meeting about my research. I thought it went well, but I didn't really know where it would go. And Alan said to me, write me a proposal and dream big. I love that. Dream big. That's my favorite command. So I did. And what Alan did was turned around and invited a number of friends of the university, so friends and alumni of Georgia Tech. And I met with them. I probably had a dozen meetings talking about my research with different folks who were interested. And many of them joined together to give a gift to the university. And that was huge because it is one of these high risk kinds of uses of funds. So the NIH is very conservative. You can only do studies that are really, the NIH only funds studies that are really, really likely to work. And you have probably already done half the study to prove that it's going to work. But this is funds where we can go after those big risky ideas. And one of them being getting this into humans. So that was huge. And then in addition, Alan said this to me over the weekend as well. What do you need? Are you having any problems? What can I do for you? And so whenever I ran into a hurdle with figuring out how to make things work in the university, if there was a bureaucratic hurdle or whatever it was, Alan wanted to know and he wanted to help. And he would get on the phone with whoever he could to help us, jump over all those hurdles that are involved in doing really interdisciplinary, cross-university, mice to human studies. I mean, it's a big leap. And so his support has been not just in the research as a donor, but as a member of the community who can energize people to get the work done.
That's great. So these gifts have really been to those de-risking of those studies, those critical studies to take it from high risk to lower risk to be able to apply for the longer journey from the National Science Foundation and National Institutes of Health. That's great. So this is a long journey. We don't have cures around the corner. And I was wondering what hope you can offer to the people on the call before I open it up to other questions.
Yeah. It is a long journey and it's hard, especially I think it's hard because we don't have a lot of treatments. We have no truly effective treatments for Alzheimer's right now. And the pipeline, there's a lot in the pipeline, but the pipeline takes a long time. So I think there's a couple different levels of hope. One is there has been a huge explosion of knowledge about Alzheimer's and research into new kinds of therapies. So for a long time, everyone was focused on anti-amyloid therapies, but there's now an appreciation that the disease really has multiple levels, multiple facets, and people are trying to target all of them. So there's a lot of creative research and translational research that's going on right now that I think has a lot of hope involved. And the other for us in particular is we're really hopeful about the results we have so far. Our human studies were, excellent in terms of the feasibility of getting people to do this. And then we have some really interesting preliminary biological effects that indicate we're seeing similar things in humans like we saw in mice. So that tells us to keep going, and we will be keeping going. So it opens up the door to doing much bigger, longer studies in the future.
Thank you. So now I'll switch over to questions, and we've had some questions in the chat. And while you're thinking about your questions, I encourage you to use them, the chat for that. Remember, it's up at the top. There's a little kind of a box like that you would see in a cartoon, and you can click on that and write your message. I'm going to share a question that was from Jack Fossman, and he had sent this ahead of time. Invitees are asked to send questions ahead of time. And I'm going to actually kind of paraphrase Jack's question. When someone's a smoker and they're asked, they successfully stop smoking, they actually have stopped the progression of their disease, if you will. And they're basically not reversing their outcome, but actually staying put at that many years of smoking, for example. So in Alzheimer's disease, do you think that the flicker therapy is something that can actually reverse or just halt? In other words, when you ramp up the garbage collectors, do you think they're actually going to be an improvement or just like stopping smoking, staying where you're at?
That's a great question. Okay, there's like multiple levels to this answer. So first of all, it probably depends on when we deliver the therapy. So in Alzheimer's disease, there's multiple stages of the disease. Late in the disease is when we see neuron loss. That is very hard to reverse. Early in the disease, there's what we call brain atrophy, but it can be due to synapse loss. So those are the connections between cells, and that is reversible. And even earlier, before synapses are lost, we see changes in neural activity and immune signaling. Those are also reversible. So if we hit at one of the sort of earlier stages before we're getting to neuron loss, I think the disease is possibly reversible. And then if we're talking about flicker in particular, so one of the biggest predictors of cognitive decline is synapse loss. So those connections between neurons, they basically disappear. So key to a therapy is probably bringing those back. And one of the things that we're seeing with flicker stimulation is that we can see an increase in synapse marker expression, or it's a measure that's related to the number of synapses. So I think if we do it in the right way at an early enough time point, we could potentially not just halt the disease progression, but reverse. All that said, our animal studies show we're able to reverse some of the cognitive impairment and reverse some of the amyloid buildup. So all of that supports that idea as well. I think in humans, the key challenge will be getting participants early, getting people early in the stage.
Mm-hmm. And identifying who is at a very early stage when the symptoms would be extremely subtle. Exactly. So there's one quick question, and then I'll turn it over and allow Barrett to close us out. There was a question about, in a similar sort of way, you're trying to restore those synapses with a traumatic brain injury. Very quickly, do you think there's potential for using flicker in traumatic brain injury?
Yes, absolutely. It's something Levi is a collaborator, he's another faculty member, and I have talked about. He looks at immune signaling in traumatic brain injury, and when he saw our effects that we got with stimulation, he was like, "Oh, I could look at these stimulation effects and my traumatic brain injury effects and figure out from the two how can I map the right kind of stimulation onto traumatic brain injury." We haven't tested any of that yet, but theoretically, there's some really interesting potential.
And there's one quick question about, have you tested where you stop the flicker and then continued studying the animal? Is there a persistence in the treatment?
Yes. So if we stop the flicker and test them a week later, a lot of the benefits are lost. So these mice, the molecular pathology continues. They're genetically programmed to continue producing the disease pathogens, and that keeps happening. So we think that as a treatment, you would have to use this every day and keep using it.
Yep. So it's like your daily workout. Exercise is good for you. Flicker. Okay.
Yes, exactly. Exercise is good for you.
So I want to thank you so much. You've been such an inspiring speaker and on such an important topic that really, quite frankly, affects every single person on this call, whether it's someone that they know or someone that's a close family member. And your work is really so very exciting and so very different than developing a new drug treatment. And it's so, we're grateful that you have shared your story with us today. Thank you very much. Barrett?
Thank you.
I want to thank Professor Annabelle Singer and Professor Susan Margulies for joining us today in Conversations on the Hill. For those who missed a portion of it or would like to see it again, please know that we will be sending out to everyone here on the call a recording of the event. As well as information about how to follow up with Professor Singer and so on. So again, thank you for joining us. Thank you to members of the Hill Society and Foundation and the Georgia Tech Advisory Board for being with us. I want to remind you that the next invitation will be coming out momentarily for conversations on the Hill. Cybersecurity will be the subject on March 23rd. Cybersecurity, March 23rd. Our panelists will include Phyllis Schneck, as Chief Information Security Officer at the Northrop Grumman Corporation, Admiral Sandy Winnefeld, who's here on the call with us today, former Vice Chairman of the Joint Chiefs of Staff, and Charles Isbell, Dean of the College of Computing here at Georgia Tech. With that, I'm going to bring the afternoon to a close, and think positive, test negative, and drink Coca-Cola. Take care now. Thanks, everybody. Be safe.